by each other's side. But tonight I feel like I'm sleeping with another woman, and you don't even seem to recognize that while you stay shut up inside like you're keeping secrets, got something to hide. I'm out in the world getting high on life, so when I come home late, you know, you bring me down. And I know that this life isn't what you thought it would be, but I care for you, baby, and it's clear to me that you'd be better off alone. Well, I've been trying to meet you, to reach out and bring you in from outside, but you're so far away, I just don't know how to keep you satisfied. Yet tonight, if it keeps on raining, I'll hold this door open. She writes, darling, we haven't been lovers under the covers for years now, since you let me down. And I don't know how you claim to know me when you're never home to hold me on those cold and lonely nights. 'Cause while you're outside getting high all the time, I'm stuck at home being somebody's wife who promised to care and to always be there. So when you come home drunk, you know you've let me down. I don't know exactly. It wasn't that long ago, before the first mobile.
Good evening, everyone. Welcome to Pint AU Online. It's fantastic to have you tuning in here tonight for an amazing panel that we've got lined up for you. I've got three brilliant scientists who are joining me online as I'm joining you to share some of the amazing work they're doing as we're going to delve into our beautiful mind and explore brain health in our modern world. So welcome to all of you who are viewing on YouTube, those of you who are viewing on Facebook. You're going to be part of this tonight. You're going to join us for this journey, and you're going to be asking lots of questions and taking part in this. You can type that in as we go through YouTube and Facebook, just underneath the video stream, or on Twitter with the hashtag PintAUPanels. So it's going to be a fantastic night for us all as we start to explore our brain. Now, Pint of Science, traditionally, when we hold events at pubs across Australia, we do ensure that we recognise the local Aboriginal and Torres Strait Islander nation, one of many that makes up the continent that we now call Australia. Now that we're holding this event online tonight, and our speakers and much of our audience are streaming from across Australia, we want to acknowledge all traditional custodians of country, the first innovators, the first scientists of this land, and we pay our respects to Aboriginal and Torres Strait Islander elders past, present and emerging, and we recognise their continuing relationship with the lands, water and culture. My name is Broderick Matthews. I'm a science communicator, and I'm super excited to be with you tonight to bring this panel to you. As I said earlier, we're delving into our beautiful mind, the topic, brain health in our modern world. And the brain itself is a pretty impressive squishy blob inside our head. Scientists have been studying it for years, but there is still so much more to discover about how it works amongst all the grey matter, brain cells, the 86 billion neurons. We don't 100% understand what's going on, which is great for us, I reckon, because there's so many different things that we can discover. And that's why we have three scientists with us tonight who are going to share some of their research and their discoveries about the brain and really tell that to us. It's a fantastic night in store. Tonight, we're going to be asking questions like, how does stress affect our brain? Can we feel better by changing the buildings around us? And why do we get diseases when we get old, like dementia, as our brains start to age? You're going to have the answer to these questions, and you'll be able to ask your own questions too through our live streams on Facebook and YouTube, or by tweeting us using the hashtag PintAUpanels. I strongly recommend you ask your questions as we go, because there's I hate it when you forget the question, so write it down as you go, ask it. And there's a fantastic Pint of Science team in the background who are ferreting away, who are going to pass those questions on to me so I can ask them to the three scientists for you. So please ask lots of questions, and it's going to be a fantastic night. Now, I've mentioned the three scientists a couple of times. I think it's time to actually meet them. So let me take an opportunity to introduce them one at a time to you tonight. Our first scientist is Maddie Hinwood. She's a research academic at the University of Newcastle, and her work in psychiatric epidemiology helps improve our understanding of stress-related disorders. Her work also improves our access to medication by recommending effective treatments for the pharmaceutical benefits scheme. G'day, Maddie. How are you going?
Good. How are you?
I'm going really well. Fantastic to have you with us, joining us from Newcastle. You excited to be sharing a pint with our audience tonight?
Yes, very exciting. It'll be lovely.
Awesome. All right. Well, we're going to get to your talk in just a moment, but I should introduce the other two scientists first. So the next scientist on our panel this evening is Bella Bauer. She's a PhD candidate from Deakin University in Melbourne. Now, Bella has a background in architecture, which you may not think relates to the brain, but I'm sure she's going to tell us why it does in just a bit. And she holds the inaugural John Paul Eberhard Fellowship from the Academy of Neuroscience for Architecture Research in Environmental Psychology. G'day, Bella. Welcome to Pint of Science.
Hey, Brod.
I'm presuming you've set up the room behind you tonight to make it ideal for communicating your science?
Yeah, the surf is going to be part of my presentation.
Awesome. Well, I look forward to hearing more about the surfer and the built environment around us. Thanks very much for being here.
Thank you.
And our final scientist joining us tonight is Luke Wiley. Luke's a senior lecturer at Murdoch University in Perth, and he's looking at dementias such as Alzheimer's disease, which are on the rise, but there's still a lot we don't know about the disease. So that's Luke's day job as a researcher, but outside of his day job, he also kind of does some work on the Alzheimer's as well. Last year, he ran the London Marathon and raised money at the same time for dementia research. So, Luke, welcome along. There's not many scientists. Good to have you. I feel like there's not many scientists who raise money for their own research outside of the office. How did you go on the marathon?
Oh, it's a fantastic experience. So at the time I was working for the Dementia Research Institute over in London, and the key partner of the marathon last year was the Dementia Revolution charity, and they were looking for scientists to join them in the running team and to help promote the great work the charity does. And it was the marathon itself was such a great day. The crowds were amazing, so I really enjoyed it.
Awesome. Well, we'll look forward to hearing maybe not so much about your running, but some more about your research later on tonight. So thanks very much for being here. Now, Pint of Science is an event that's held in a pub traditionally, because a pub's a pretty relaxed sort of place, and it's a place you can chill out and just enjoy hearing about this amazing work that our researchers do. This year it's gone online and you're in your homes, but I'm hoping you're feeling relaxed. You've got your Ugg boots on. You might have a pint yourself as we go through. And so I hope you really are ready to just sit back and enjoy our speakers tonight. I'm going to hand over to our first speaker now, Maddie Hinwood, who's going to talk about stress after stroke and does it affect recovery, all this stress. So let's hear from Maddie now.
Okay, thanks, Brod. I'm an early career researcher at the University of Newcastle. I'm a neuroscientist and epidemiologist. So I'm going to tell you a little bit of a story now about how I came to be where I am now. I finished my PhD in neuroscience in 2013. During that degree, I was looking into the way that the brain different cells in the brain respond to chronic stress, and especially the role of the brain's own immune cells, which are called microglia. Chronic stress is among the most common antecedents or precipitating factors to a lot of mental health problems, particularly anxiety disorders, depression, other mood disorders, and post-traumatic stress disorder. So the studies that we did in this lab while I was doing my PhD provided some of the first data around how important these microglia are for responding to stress and maintaining brain health. In the years since then, this has become part of a really large body of research showing that chronic stress has major impacts on the brain and behaviour. In the last decade in particular, what we know about microglia and the immune system and the role that they play in regulating neurobiology and behavioural outcomes has really exploded. After I finished my PhD, I took a little break from research and I worked in public health for a little while. And while I was working in public health, I fell in love with epidemiology. So I started a Master of Clinical Epidemiology, which I've only recently finished in 2018. Epi can be a difficult science to explain. So I was looking it up for you to help explain it. And last year, there was a really beautiful article published in the American Journal of Epidemiology, which described it as being the quantitative heart of public health. And I completely agree. Epidemiology is the science of the health of populations and the processes that shape that health. So epidemiologists use a lot of different methods and data sources to try and understand what causes health, both good health and poor health. So at the moment, I'm a research academic in health technology assessment, a member of one of the academic teams across Australia that provide advice to the government on effectiveness and cost effectiveness of new health technologies. And at the same time, I'm establishing my own research career in epidemiology, and that's what I'm going to give you a brief overview of tonight. So at the moment, there's in research, there's a lot of talk about research translation. And in health, this means bringing your research through to the next stage and hopefully to the final stage, which is where it's somehow being used to improve the health of people. So when I was in that preclinical lab doing my PhD, I had a very clear narrative in my head about how we were beginning to understand about the way that stress changed all these cells in the brain and how that would eventually lead to new treatments for stress-related disorders and help people. But really, in spite of all these decades of knowledge about how bad stress is for us, we're still not really seeing widespread stress management being advocated in human populations. I think research translation is really, really hard. It's really hard to do well. And now that I've worked in both the very start of that research pipeline in the lab and also at the very end, which I would say is health technology assessment, which is the final stage before a new intervention is available to the population, I can see how hard it is to bring things along this pipeline to where people can use them. So ideally, what I want my work now to do is to try and help this, help stress come along this pipeline and try and raise awareness about stress in human populations, by which I mean trying to intervene in stress and to prevent the cognitive and psychological problems that people can experience following exposure to chronic stress. So in trying to identify what might be important for clinical stress research, ideally, as stress is a major antecedent to these mental health problems, maybe there's a chance to intervene when people are experiencing stress and try to prevent some of these mental health problems from occurring. So I'd like to ideally do for stress what a lot of fabulous public health researchers have done for the prevention of other physical illnesses like cardiovascular disease and diabetes with other behavioural risk factors like physical activity and nutrition. So we want stress to be recognised as an important risk factor for poor mental health and to maybe try and come in at stress at a preventive level before these disorders can develop. So one way we can do this is to recognise that there's populations of people who are at risk of stress-related disorders, and people who have experienced a stroke are at risk for stress-related disorders. And we know this from two separate sources. Firstly, several studies have now shown that stroke survivors report experiencing high levels of stress than the general population. And secondly, stroke survivors also experience increased rates of stress-related disorders compared with the risk in the general population, particularly depression. So, for example, in the 2 years following a stroke, the risk of depression is around 25% compared with about 8% in a control group of people the same age. So that's more than triple the risk of depression for stroke survivors. So a lot of these studies measuring both stress and stress-related outcomes have been done in the first 6 months to 2 years after stroke. But at the same time that we've been increasing our knowledge of stress, stroke researchers have developed these amazing treatments for stroke in recent years. So with both clot-busting drugs and clot retrieval technology vastly improving survival rates, which is excellent. It means that more and more people are surviving stroke, but it also means that people are living longer with potential ramifications of stroke. Another thing that we know is that the recovery trajectories of people following stroke are really different. And the difference in recovery is not always explained by the severity of the stroke itself. So what might be some of the factors that contribute to the differences that we see in stroke outcomes? So given what we know about stress, that chronic stress is associated with both a biological response that negatively affects our brain cells and can cause also negative cognitive and psychological outcomes, and that stroke survivors are reporting experiencing high levels of stress, maybe stress is one of these modifying variables that changes the outcomes of stroke recovery for people. So our hypothesis in this study is that the chronic stress experienced by some people after stroke will lead to both poor brain repair outcomes. So after stroke, the brain is capable of an enormous amount of repair on its own, but the negative effects of chronic stress is likely to interfere with the repair processes of the brain. Also, because people are feeling stressed and maybe feeling some of these negative behavioural consequences, they might be engaging less with rehabilitation services, which in turn could create more feelings of stress and lead to a cycle whereby people feel more stressed and engage less with services. So we've run recently a study looking at a group of people who had had a stroke who were now living in the community at any time after they'd had a stroke. So we didn't limit the period of time sort of to that first 2 years. We looked at a very mixed population, and our study included participants who had had a stroke 6 months ago out to 25 years or more. And all of these people, on average, reported a high level of stress in an age-matched population of controls, so people who did not report a previous stroke but were the same age. And interestingly, the relationship was maintained regardless of how long ago the stroke was. So on average, even if the stroke was 25 years ago, the people in that group were reporting more stress than controls of the same age, which suggests that for some people, these feelings of stress might continue for quite some time after they've experienced a stroke. We've also showed that for people who had a stroke who reported experiencing higher stress, this was correlated with their cognitive and psychological problems in areas like mood, thinking, and memory. So stress might be an important factor associated with differences in stroke outcomes that we see. So now we're thinking about what to do next. Maybe addressing stress will help to improve rehabilitation outcomes for these psychological and cognitive problems that people can experience after stroke. So this includes both looking at potential sources of stress after stroke. The source of stress might not all be due to the stroke itself, but also unmet needs experienced during rehabilitation, and also working out what is the best way to tackle stress. So what intervention is needed. So that's where we're at now, trying to raise the profile of stress as a behavioural risk factor, maybe develop the right intervention for it, and also look at people who are resilient to the effects of stress as well as those who are vulnerable. We just don't know yet what's the best way to tackle this. And I also wanted to say thank you to the amazing team I work with. I have a lovely group of mentors and other researchers who work with me on this project. And I've met some really lovely people doing this work, including our wonderful consumer investigators. So thank you.
Awesome. Thanks so much, Maddie, for sharing the work that you're doing on stress and stroke. And folks, if you're listening at home and you've got some questions for Maddie about chronic stress or how stroke is affected by it, or maybe how you can stress less, prevent some of it, then make sure you ask those questions on our live feeds or tweet us at #PintAUpanels, and we'll get all those questions together to start asking our panelists in just a little bit. But thanks for sharing that one, Maddie. I'm going to throw over to our next speaker now. And next up on the list, we have Bella Bauer from Deakin University. And Bella's going to be sharing her talk entitled, "Do Buildings Affect Your Emotion?" Let's hear from Bella.
Thanks, Brod. So I'm a third-year PhD candidate at Deakin in the School of Architecture and the Cognitive Neuroscience Unit with Richard Tucker and Peter Endicott. I have a bit of a different background. I came from architecture. I did a bit of time in architectural practice. I then moved into government, so I looked at built environment policy in the Office of the Victorian Government Architect. And then I went into research and decided to pursue a PhD. So to start off my talk, I'm going to ask you to think back to a significant moment in your life. It might have been getting some medical news, so waiting for some test results, or sitting a really important exam, so perhaps your VCE results or something like that. And I want you to think about the building and the space you're in. So what if that building was affecting your emotion? That's something we just don't know at the moment, and that's my research question, and that's what I want to answer. So we spend about 80% of our lives inside buildings, a lot more considering the current situation at the moment too with COVID. We've got rapid inner-urban environmental growth, rising mental health issues in society, and a lot of money goes into built environment infrastructure. So what I've developed is a technique that I'm testing at the moment to measure our emotional response to buildings. If we go to the first slide, I just want to put up a bit of a provocation, which is that we have buildings where we're making sure they're physically safe so that we don't slip, that we don't fall off a balcony, but we're not considering how they're affecting us in a psychological manner. Now that's really tricky. You can't see how a building affects you. You can ask someone how they feel in the space, but that's very subjective too, and that relies on that person being able to tell you how they feel. What I cross over a little bit with Maddie and Luke is a theory called environmental enrichment, which I think we'll cover a little bit of tonight. So it's a theory in neuroscience where you have an animal in quite a plain environment versus one in an enriched environment. And that enriched environment will have a lot of different elements. It'll be about the diet of the animal, it'll be about social interaction, it'll be about the other objects in that space too. What my research is trying to do is see if architecture is actually one of those components of environmental enrichment to then measure that effect. So the other important thing to say with environmental enrichment is that when you ran experiments where the animal was in a plain condition versus enriched condition, there was cellular, molecular, and behavioural changes. So not only was it affecting performance and abilities, it was also affecting the brain chemistry and structure within the animals. Now, it's really interesting to go, right, if that's happening in animals, is that happening in humans? It's quite tricky for us to control things in human studies as much, but what I'm trying to do is control that. So I can see we've gone to the second slide. So how I'm doing this is basically a person is in the middle of a virtual reality space. So instead of thinking of the traditional headsets where you've kind of got this cover over your eyes, I used a virtual reality cave, which is a computed automated virtual environment. It's basically like standing in a space with projector screens around you. It's really cool. You wear glasses, and so your vision flicks really quickly, which gives you that illusion of depth in front of you. I put sensors, so electrodes, on people's scalps using a technique called electroencephalography to measure brain waves. I'm also looking at how the body responds. So I'm looking at sweat response and heart rate, and that's to see if you're having a response before conscious awareness. I'm also asking people how they feel. But what's really important too is that I'm controlling for comfort variables. So very little studies across the world have looked into how buildings affect people, and those that have done this have used very small sample sizes, and they haven't reported all the conditions, so it's very hard for us to reproduce them and understand truly how it affects us. So what I did was I measured what the temperature was. I made sure I was looking at the acoustics, the airflow, the humidity, and I was doing that at the level that the person was at. So it's important you also measure this at the head height versus the torso versus the feet. The other thing is the built environment's extremely complex. So part of using a virtual environment was that I could really control it, and I could control what I showed the person, whereas a full built environment has a lot of things going on in it, basically. In this diagram, what you'll see is a few little symbols. So what I was looking at is the colour of the space versus the texture versus the scale versus the form and geometry in that space, to try and break it down into really simple visual elements, so then we can hopefully see if there is an effect there, see if the method itself works to measure emotional response, and then build up complexity and understand how that does affect us. So I tested over 60 people. I'm still analysing the results, so I will be reporting back this year as to how it does, what the results were, basically. And I did have a really great moment, actually, where it wasn't great at the time. I was very stressed, where a moth came into the cave, and because it's dark, the dark VR room, and you have projector lights, it was attracted to the lights, and so it landed on the light, and there was a huge moth. My experiment, I was very stressed at the time. Then I ran with the lab tech to try and catch the moth after the experiment to get it before the next participant was like, why is there a big moth in my... Yeah. Anyway, we got to the bottom of that. It's very hard to control everything in science, though, even though you really do try. So what's the importance and what are the future implications of all of this? Well, we want to know if we can use buildings to have a therapeutic effect on us. Like, what if we could use the built environment, the spaces where we live, we work, we heal in hospitals, education, schools with developing minds. What if we could make those environments have a positive effect on us and make sure that that was helping us? Architects and building designers, they have a really important job. They're creating all these spaces around us, and we really want to say what good design is, but it's very hard to at the moment until we can actually say, well, it is having an effect or it isn't. And it's just something that we haven't been able to do until now with technological developments. It also raises some ethical questions in a sense, because if buildings are having a negative effect on us, well, we don't want that. We want them to have a positive effect and help us in our lives. So future questions, well, I want to build up complexity. The built environment is an experience without other people in it, whereas what I've done is controlled and not put other people in the space because perhaps you don't like that person in front of you, perhaps they smell a little bit. They're all kind of confounding variables, and until we have a baseline, we need to get that baseline first and then build complexity. I'd also really like to know the thresholds of how design elements affect you. So is there too much colour? Is there too much of a scale? Is there too much of these interactions? And how would the sensory input change? Because what I'm looking at the moment with scale and colour, that's a very visual sense. But if someone was blindfolded and they were just relying on auditory cues about the space, as to what the scale of that space was, seeing all those signals are actually just broken down into electrical messages, would it be the same emotional response? So that's basically my research. I hope you have some questions for me, and thank you.
Thanks, Bella. And I can see the questions coming in already from our YouTube and Facebook channels. So people are definitely asking lots of questions and tweeting us too with the hashtag PintAU panels. So if you do have more questions about Bella's amazing cave, which I kind of want to have a go in, it sounds like a really cool show ride, if I'm honest. If you've got questions about that or the built environment and all these things that Bella's trying to measure, then please ask those questions and we'll ask them of Bella in just a moment. Let's throw to our final speaker now, though, and that is Luke Wiley from Murdoch University in Perth. And Luke is going to be answering the question, is dementia in our genes or in the way we live? Let's hear what Luke has to say.
Yeah, hello. So good evening. As Brad was saying there, I researched the reasons behind why and how someone may develop dementia as they age. And what do I mean by that? So first of all, if we take the term dementia, it's a term that covers a wide range of conditions, which are typically associated with some form of cognitive decline or cognitive challenges. Now, the work I'm going to talk tonight about is a type of dementia, Alzheimer's disease. Alzheimer's disease is the most common dementia. And the biggest risk factor that we know about Alzheimer's disease is age. The older you are, the higher the chance you have of developing Alzheimer's disease. Now, inside someone's genetic code, we also know there are specific risk genes that may increase your chance of developing Alzheimer's disease. But just because you carry one of these genes doesn't necessarily mean you'll go on to develop Alzheimer's disease. So there's obviously more at play here. It's not simply written in our genetic code. Of course, that does play an influence, but there are other things throughout our lifetime that are likely to influence that process. And perhaps it's in our dietary choices, our lifestyle choices, whether we exercise a lot, whether we eat healthy diet, smoke, drink, it's all going to have influencing on our overall lifetime health and likely our brain health as well as we get older. Now, how do I study this? I use a technique called metabolic phenotyping. Now, how this works is we measure blood samples and urine samples collected from the general population who are partaking in health studies. And we look to measure as many chemicals in those samples as possible. We use some very powerful analytical chemistry technology. I don't know if any of you watching this were also watching on Monday night when Ray Ling was explaining about how she can spot the type of food that someone eats from their urine. We do very similar work, except we're not always necessarily looking for food metabolites. Perhaps we're looking for markers of bacteria or markers of stress, infection, and all of this combined together gives everyone a unique a fingerprint, if you like, that we can compare together. Now, if we get enough people and we analyze enough samples and we know from that group what the outcome's going to be, we can start to look at patterns in that data. So maybe the group who in later life develop Alzheimer's disease, perhaps they have an elevation of chemical Z during their life. Now we can find out where that chemical came from, and then we can start to picture the pathway that perhaps leads to that person developing Alzheimer's disease. Now this is really important because if we can pick that up early enough, there are multiple benefits here. Perhaps we can diagnose Alzheimer's disease earlier. Perhaps we can intervene earlier and develop new treatments for the disease. And perhaps we can, by treating it earlier, we can slow it earlier. So therefore, the effects are seen later. So by analysing this chemical fingerprint at various points throughout a person's lifetime, we are aiming to spot patterns that perhaps are linking our genetics, our environment, our lifestyle all together in order to see which of those are the most powerful drivers of an individual developing a disease later as they age. So in a summary, that's kind of the work we do here. I work alongside a centre called the Australian National Phenome Centre. It's a new centre that has been in operation for 6 months, and it specialises in generating this type of data from tens of thousands of samples. And the idea is if we can get enough of these samples from large health studies, we can start to spot real subtle patterns in the data, and then we can get a really good idea, a really good indicator of what those chemicals and those metabolites are doing to our metabolism through our lifetime, which actually results in cognitive decline as people age.
Fantastic. So much for that. It's amazing to hear. And it has been to hear all of our scientists' research and the work that they do through that. So again, keep asking those questions, folks. I can see them coming through on all our scientists there. And as we continue to ask these questions of our panelists tonight, please keep asking them yourselves so we can keep going. I'm going to throw back to our first speaker, Maddie, to ask a couple of questions about the stress and strokes that you were talking about earlier. And there's a couple of different people asking similar questions here about whether chronic stress is the cause of the strokes or what's actually going on. So Crispy Chicken on YouTube asked, do you know if the chronic stress is because of the strokes, or could the chronic stress have contributed to the stroke, resulting in, kind of a biased sample that have higher stress levels?
Yeah, so the answer is a bit of both. So there's been some fairly famous epidemiological studies where they've looked at people who were stressed and measured the rate of stroke in those people over time. So one example, like stroke and other cardiovascular disease. So I think one famous example is that during the World Cup, one of the World Cup finals in Germany, people were at a high risk of having a cardiovascular event while they were watching that grand final because of the stress that they're experiencing watching their team try and win. So yes, stress is definitely a risk factor for any cardiovascular disease, including heart attack and stroke. But what we're interested in is the way that stress after stroke affects stroke recovery and stroke recovery outcomes. And I mean, it's difficult to sort of pull these things apart. That's what EPI is trying to do. We're trying to work out what is the cause of disease. And I think the answer probably is that we just don't know yet exactly if, it's a high stress population that's more prone to cardiovascular disease, because there's a lot of different risk factors that contribute. But yeah, the area that we're looking at is the way, and not everyone is stressed after they have a stroke. And that's, something that we're looking at there too, the individual differences in the way that people are feeling stressed and the impact it might have on their recovery trajectory from stroke.
Okay, and I guess the one question we should probably clarify too, which a couple of people have asked the question on, Elise Kenny on YouTube and Lisa McKenzie on Twitter ask, how do you actually define stress? And how bad does it have to be for, to be chronic stress to become an issue?
Yeah, that's an interesting question too. So in our study, we measured stress a couple of ways. So the result that I was talking about was using the psychosocial stress scale, which is like a psychometric scale that people have developed to measure how stressed someone's been feeling over a certain period of time. And it's a very popular measure to measure stress. You can look up exactly what the questions are online, I think. And there's a few of those. I think the PSS-10 is the most popular measure, but we can also look at biomarkers of stress. So the stress hormone that's released into your bloodstream is called cortisol. It's tricky to measure in the blood because as well as being released in response to stress, it's also has these peaks and troughs through the day as part of your natural diurnal cycle. So if you're going to measure that in a study, you tend to have to measure it at the same time every day and probably a couple of times a day in your participants to get an idea of what their own baseline is. As well as then how stressed they might be feeling. And probably another point that would be good to make on that question is to talk about the difference between acute and chronic stress. So an acute stress response is an adaptive response to an acute stressor. And I had a lecturer in psychology talking about this when I was doing my undergraduate degree. And I can remember her talking about, when we were, maybe it's psychologists, they like to talk about the way that our reflexes and things would have helped us survive as cave people. And so the acute stress response would have been a response to save us from an acute attack, like running away from a tiger, or it's also released in response to a serious injury. And the aim of that stress response is to sort of try and shut down non-essential processes and prepare you for that flight or fight mechanism. So it's quite adaptive to an acute stressor, but when we're experiencing it day after day, shutting down those, non-essential things for responding to fight or flight includes things like a digestion and certain parts of your circulation. So that, I think this is when you start to get problems when you're experiencing it day after day. So
yeah, and it's quite interesting when you start applying those, very primitive, I feel like is a bad word for it, but those primitive fight or flight responses, which apply to us many, many generations ago to the world we live in now. So it's quite interesting to look at stress in that way. And I guess, the different people respond in different ways. And speaking of different people, Bella, we've had a few people ask with your work, does it affect different people in different ways? For example, Sarah Morecambe asked, does it affect people, does it have changes based on age or culture? Fruit Salad on YouTube asked if you were looking at a neurodiverse sample. And Shweta Joshi was asking about the age or gender of the subject. What differences do these sorts of things have to the responses you see?
Yeah, absolutely. That's something really important to, again, try and control for and ask to start with. So I use the Big 5 personality test to look at people's personality who completed my experiment. So you can look that one up online. That's the IPIP-NEO, and I use the abbreviation abbreviated version of that because it was a little bit shorter. But also, while I did decode my participants, I did look at the difference in gender, in where you grew up as well. So you might have grown up in Hong Kong, in quite a high density city environment where there's multiple layers to the city, but also that, you know, you're in a small apartment versus growing up in rural Australia where you're in quite a large expanse too, and what difference that would make. These are all great questions, and these are all things that it's why I ask those questions, just to see when I do decode my results as to whether that did make a difference or not. So yes.
Yeah, awesome. Well, and moving on to you, Luke, some people have been asking a similar sort of question around your research. The Bubs on YouTube asked, you know, apart from age and genetic markers, what are the biggest risk factors for types of dementia? Do we see it occurring in different rates in different people?
Well, so this is a lot of work still going on in this area, particularly epidemiology. As Maddie was saying, as that Maddie works in, they're looking for patterns in lifestyle and what happens. One of the big risk factors that is potentially showing itself in the research is cardiovascular health and diabetes and obesity, and increases in dietary habits, or changes in dietary habits, I should say, as populations change. And that is becoming quite apparent that their overall body health and cardiovascular metabolic health is having a negative impact on the brain. There are some hypotheses that are suggesting that perhaps it could be damaging the blood-brain barrier somehow, so in causing inflammation in the brain. But a lot of this work is ongoing in that area, but certainly sort of general overall health and throughout someone's life is showing itself as a risk factor for dementia as we age.
Mm. And speaking of all those samples, Luke, that you were mentioning there, someone else has asked us, how do you actually study this metabolic phenotyping? Do you get brain samples from people or cadavers?
So a lot of our samples that we specifically look at are actually in healthy controls who are young adolescents or even middle age, because we want to track how metabolism changes with time. So if we are collecting samples from people who have already been diagnosed with dementia, we can use that potentially as a flag to see if any metabolites are changing. But the likelihood is that they're already being diagnosed drugs and treatment and therapeutic interventions, which in turn are editing their metabolism. So it's very hard to get a true signal from what's being caused from the disease. So if we can track samples over time in what we call aging cohorts, where people volunteer and return and give multiple samples every year or 5 years, we can try and track changes of those metabolites over time. And then we can do follow-up studies, and we can see how many of that population developed dementia longer term. And perhaps we can then roll back through the years and through our samples and see, actually, this signal was strong in that group. Was that having an effect, which later then manifested with a diagnosis of dementia or caused the underlying symptoms? So it's actually very interesting to look at that middle-aged population and to look at their health trends, their environment, and also their genetics, and see if we can pick patterns at that stage before a diagnosis of dementia has been made.
Yeah, and you're talking about taking samples over time to find those metabolites. So you're not taking samples of the brain, are you?
No. So again, because once an individual has passed away, the likelihood is their metabolism changes quite a lot. So it's quite hard to then track that back into a blood sample or a urine sample to see how that has changed before that. So we mainly work with blood and urine samples. Another really interesting biofluid that we like to work with, but it's hard to use, is cerebrospinal fluid. So the fluid that sort of surrounds our brain and our spinal cord, because that is an interchange between blood and the brain. So it was almost like a reflective fluid that we can use to give us sort of a bit of a close example of what is going on in metabolism in the brain at that point in time. So we try and get a collection of different samples, but typically blood and urine, because they're the easiest to collect, and therefore we can get them in high enough numbers to spot these subtle patterns. And I think getting those high numbers are quite important, because there is no magic chemical that we can see and say this person is going to develop dementia. It's more like the combination of chemicals and pathways and interactions in our body. And it's not only our metabolism that's influencing this, there's also, we live with millions of bacteria on our skin and in our stomach and in our gut, and they're all having into play as well. So you can imagine a real complex web that's being woven. So it's likely to be a pattern of chemicals rather than one specific marker.
Yeah, and another viewer online has asked, what age do we start seeing these markers, this pattern of chemicals appearing?
So the latest thinking is that dementia, or I'll say Alzheimer's disease, the mechanisms that start to cause the disease probably appear around 20 years before we start to see symptoms. There's a very, very long pre-phase before we can actually see signs and symptoms. And that's why these marker molecules are so important, because diagnosis is currently only really made when someone presents themselves to their doctor and complains perhaps of some cognitive difficulties. And there's a range of cognitive tests that they may take, and then there's a diagnosis by a clinician. But therefore, it can be quite challenging to interact with that process, particularly with therapeutic drugs. If we can get that diagnosis earlier and earlier, then perhaps we can intervene earlier and really slow that that disease progression. There was a recent scientific paper that came out last week, 2 weeks ago, where they're actually looking at proteins in the blood, and they developed a new blood test, and they validated it in 4 different cohorts collected from 4 different countries. And that really gives us hope that there's going to be an earlier blood test for Alzheimer's disease coming in the not too distant future.
That's some really promising results there. Speaking of blood tests, Maddie, we've got Madeline Bullock, another Maddie, asking on YouTube about the stress and whether it could be something that we can detect as a genetic phenomenon, or is it more something that's biological or mostly due to environmental factors? You know, in the same situation, is one person more likely to feel stress compared to another?
Yep, I definitely think that some people are probably more prone to experiencing the negative effects of stress. So all humans would have the capacity to deploy a stress response in response to a stimuli that they found stressful or threatening. But I think some people probably have an exaggerated response, and those people are more prone to developing stress-related disorders. But yeah, it's definitely, so it's an environmental stimuli, but it's a biological response that your body's mounting to that stimuli that causes the detrimental effects when it's repeatedly applied. So, you know, if you're experiencing stress every day at work or something, that's not very good for you.
Yeah, so it is, there might be some biology involved, but it's definitely around the environment in there too.
Yeah, the environment that you find yourself in. But it's the like the link between them. Okay, there you go. Well, speaking of the environment, Bella, you talked a lot about, you know, changing the environment that we're in, and Kevin Law asked on YouTube, would decorating your room, for example, with posters of your favorite sport team, be a form of environmental enrichment? You know, what do you think?
So environmental enrichment will be quite a few things, and changing your environment itself, that is environmental enrichment too. So you've got a lot of complexity. So if you're in that same static room that has those posters, and you're just in that room because you're in isolation, well, that's not a very enriching environment. But then if you're bringing in chatting with different people and moving into different rooms in your house, then perhaps that would probably be a bit more of the enrichment. But being in the same thing is not the enrichment. Yeah.
So there's a whole bunch of questions coming in at the moment. So we're going to keep strolling through them. So you're talking about environmental enrichment there, Bella. Grant Carruthers asked on YouTube, is it scientific feng shui that we're talking about there? Is that kind of where we're going?
Well, it kind of is in a way, like design aesthetics. Some people will say, yes, this works, and no, that doesn't work. But how do we know unless we know how the brain and body are actually reacting to it? Because we can ask people how they feel, and that's very valid. And it's really important to get that detailed response from people as to how they're feeling and if that is a good thing or bad thing. But what if you're actually really stressed in that space, but you don't know that you're stressed and you're anxious in that space? That'd be really bad. So we really do want to know that, so then we can design environments that are having a positive effect.
Yeah. So can people take action themselves to have a more positive effect in their own environment?
I don't think we know that yet. So that's hopefully what my research question is going to answer. It's going to say, well, hey, does this technique work? Can we measure emotional response to the built environment, where we're looking at the autonomic nervous system, the central nervous system, and also conscious response? And then if it does have an effect, well, then that's when we need to start building complexity, because what I really looked at was just the scale of a space, so how big and small it was and how that affected someone. And I also added in some colour too. We need to then put those elements together and see how a really complex space affects you. And we also need to move it from just looking at virtual reality, where I can control it quite a lot, to putting it in the built environment. I mean, if I had a bigger budget, if I did a postdoc, perhaps, then I would really want to look at how the physical built environment affected you too, and compare that to my virtual reality results, because they're not the same. They're never going to be the same, but it's the best I can do and the best we can do to start with, to build those little baby steps and get a bit of a foundation level knowledge.
Well, and that's often the way with a lot of scientific research, isn't it? We start in a general sense, testing it in a lab, and then the ultimate application is when we can start applying it to the real world.
Absolutely.
And Maddie, we've got a question from Claire Sargent on YouTube about your research and whether your findings for stress application, for stress in stroke survivors, has any application for the general population.
Yep, I think it definitely will. So we are looking at vulnerable populations first, so populations that we know are likely to be exposed to a lot of stress. But I think, there'll be any number of those populations across, the entire population of Australia or the world who are exposed to stress of some sort. So, it's just really interesting. Stress research has really expanded probably over the last 3 decades, and we know a lot about the impacts of stress on the brain and on behaviour, but it hasn't, we don't really talk about it a lot for humans. So some work now being done in cardiovascular disease. We're doing some work in stroke, and some people do some work in cancer patients. So these are all populations who we expect are likely to be experiencing high levels of stress. But yeah, I think this work is definitely applicable to the broader population. And so it doesn't matter what the source of stress is, the biological response is the same. And if you're exposed to that stressor repeatedly, we know that that's not very good for your brain, and that it can precipitate certain disorders.
Okay. Yeah, so very interesting indeed. And Luke, we've covered a bit on Alzheimer's and some of the things you've been measuring, but Mark Milner on YouTube asks, who wants to get into specifics, have you found a specific marker or markers in the blood urine that is modulated with age in the Alzheimer's patients?
So we've definitely seen patterns, and some of the biggest patterns of changes we see are in a group of molecules called lipids. Lipids is a term that covers fats, oils, and you might have heard molecules like polyunsaturated fatty acids. It's a broad term. Cholesterol is a lipid, and there's increasing evidence to show that lipid metabolism on a whole is dysregulated in the disease. Now, is this an influence of our genes, because our genes eventually result in the enzymes and our metabolic capacity. So is it being influenced by our genes, or is it also being influenced by our environment and the food we're eating, or the stress, or causes lipid breakdown, or are we exercising? And we don't know. But there's certainly evidence there to show that there's dysregulations in this family of molecules. They're very important, and their biological role is very complicated and being studied and sort of inched out exactly what lipids do. They're involved in signaling processes, they're involved in cell membranes, they're involved in energy storage. So there's a lot of complexity behind this subgroup. But a lot of the work we do is trying to identify the specific lipids that are dysregulated in dementia as we age, and trying to work out exactly what metabolic pathways they fit into, and to find out exactly how they change and why they may lead to dysregulation in the disease.
Quite amazing. Well, it's been some amazing answers from all of you tonight, sharing your work, and I think to finish off, Pint of Science has changed this year because of COVID-19. We wouldn't be sitting here on YouTube or Facebook doing this interaction if it wasn't due to the impact that has had on our society. So to finish off for each of you, I want to ask, how do you anticipate the social changes that we're feeling due to COVID-19 are going to impact your areas of research? What do you reckon? I might start with you, Bella. How do you think this new social world that we find ourselves in is going to impact your research?
I'm hoping that people are going to take a lot more notice of their environment.
Finish. All right. I feel like it wouldn't have been one of these broadcasts if we didn't have at least one gremlin jump in. So we did jump offline there for a minute, folks. But I'm sure you've had Zoom calls over the past few weeks that you've experienced exactly the same thing. So thanks for persisting with us. I'm going to ask my final question again to all our panelists, which was, how do you anticipate that the social changes of COVID-19 are going to impact your areas of research? Bella, we're going to start with you. What impact do you think it's going to have on your work?
Yeah, absolutely. I think people are going to be much more aware of the environments they're in and how that affects them, because they've been in the same environment for a long period of time. I think people will really crave going into different environments and different buildings and seeing different people again. So I think awareness will be raised about how important it is being in different spaces.
For sure. I know for myself, I've been working in this office for the last 6 weeks, and I've been very aware of what's behind me and what's in front of me. And I've been trying to change it about to achieve my ideal office. So it's definitely having an effect, I think. Maddie, how about your work in stroke and stress? How do you think that might change?
I'm not sure specifically for stroke survivors, but it might increase the awareness of stress. I think there's a lot of studies already kicked off trying to monitor how people are feeling stressed during this time, what sort of things are making people feel stressed, and hopefully it's raising some awareness of the detrimental effects of stress. So it's something to be aware of during this time, definitely. And as I said, we're not really sure what the best thing to recommend for stress is yet, but I find it sort of helpful to try and identify the source of stress and, see if there's something I can do about it. Sometimes that helps. But yeah, it's something that we're working on. And I think that over this period of time, it's sort of coming onto more people's radar, which is a good thing.
Yeah, I definitely think mental health has been coming top of people's lists of what's going on at the moment, and stress is a big part of that. You know, looking after our brain is looking after our mental health. So, yeah, I think you're right there. And finally, to you, Luke, how do you think this COVID-19 is going to affect your work in the Alzheimer's dementia area?
Yeah, so it's going to have quite a significant impact, I think, because it's multi-levels to it. COVID's obviously caused a lot of problems, and they've been widely documented, but dementia hasn't really gone away. And what has happened is a lot of the funding priorities have shifted. So there's going to be a lot less funding available for research in dementia. And not only that, the charities are having a bit of a difficult time because the focus has been on COVID, and they haven't been able to get their message out and their support out, and they haven't been able to raise funds as they traditionally would. A lot of the big sporting events have been cancelled, which are traditional fundraisers for these charities, and a lot of them have kind of been forgotten a bit because of the constant coverage of COVID. So I think it's important that we remember all the good work that's going on for conditions beyond COVID. They haven't gone away, they are still there. And it's important that we still try and support those charities, because they also play a really important role in supporting individuals who live with dementia and provide really important support networks and social networks. And this is a particularly isolating time. So it is really going to impact dementia research as a whole, but particularly it's going to impact individuals who live with dementia and the families associated with that. So I think we should try and remember them and think of them and try and support those people who need our support at this isolating time. So, yeah.
No, it's a fantastic message. And I think that's a great spot to wrap up too. It's a good reminder there are scientists in Australia and across the world who are working to solve the problems that we face due to COVID-19. But there's also a whole bunch of scientists across Australia and around the world who are continuing to solve the problems that affect us as a society all over, whether it's stress and stroke, whether it's creating built environments that work for us, or whether it's solving the problems of Alzheimer's and dementia. And the many more scientists that we've heard across this week from the Pint of Science, Pint AU online. So look, it's fantastic that we can still celebrate that and be a part of amazing events like this, even if they are online. And I think that's a little plug. Keep an eye out for your friendly scientists and keep supporting that research in the ways you can, whether it's a positive tweet on social media, whether it's donating to programs that support them, or whether it's a friendly nudge to an MP or a politician. That might help too. But look, I'm going to take this point to thank all three of our speakers tonight. If we were live, you'd probably hear a fantastic round of applause from all the audience, but unfortunately it's a bit hard to hear that. So I'm going to give you one. Well done to all three of you, Bella, Maddie and Luke. It was fantastic to have you on board tonight sharing your research with us.
Thanks, Rob.
Thank you.
That was the last of our 4 panel discussions that we had, which is a completely new type of event for Pint AU. It's been an intense experience for everyone in this, but it's been a hugely rewarding experience too to bring you all Pint AU online. You can find tonight's earlier panel and the 2 on Monday and this one. They'll all be up on YouTube, so you can watch them again if you've already seen them, or check them out if you haven't. We also had trivia last night with over 150 teams playing online. That's a pretty massive science trivia. I must say, I did it myself with my friends. We weren't as successful as we thought. There were some tough questions out there, but there are some very smart teams too. So it was a fantastic experience. And you can also check out scientists who took part and shared their work through the hashtag #ThisIsMyScience on Twitter and Instagram. I think there are over 130 scientists that shared their work. There's some amazing science going on out there across Australia. So take a look at that. And on Reddit, we had our AMAs, the Ask Me Anythings, some scientists who were getting the tricky questions from the public, not just in Australia. I saw some questions coming through from random places like Belgium and the US too. So check out those Reddit AMAs on the Pint of Science account there. Now we've come to the close of the live events. As I said, you can rewatch these on YouTube. You can also listen to the podcasts. We have Pint AU's podcast, which is called Pod of Science. Yeah, they're short interviews with scientists working in Australia, different scientists to the ones that we've had on these panels, scientists that were all lined up, ready to speak at these live events, and then they didn't happen, so we're getting them on a podcast instead. So you can check that out. You can check out my own podcast as well. I host a local Canberra show called Fuzzy Logic, and I interviewed a couple of scientists this week. So I'll be podcasting my interviews with them, talking about astro-optics and scientific illustrations. I had a great time interviewing them. So that's Fuzzy Logic coming out this Sunday. And you can listen to my own podcast, This Was the News. It's got nothing to do with science. It's a historical news podcast, but heck, I got to plug it, don't I? In a couple of weeks, I'll also give Luke a plug. Luke Wiley from tonight's panel is going to be speaking on the podcast Curiosity Killed the Rat, which is a fun science podcast with a bit of a balance of facts and fun and a lot of banter. So you can look forward to hearing Luke on that one, sharing his thoughts and some of his work. As the final panel tonight, I'm going to take the opportunity to say a big thanks to the more than 250 volunteers and 450 plus speakers who signed up for the original pub events that had to be cancelled. Thanks so much for all your support, folks. We hope to come back to you in 2021 so we can have these events in person, in pubs around the country and share a pint together. Huge thanks also to the small team of about 20 people who've done a massive amount of work in the last month to put these new online events together. They've just been amazing. I've been thoroughly amazed by all those that I've seen, and I hope you've enjoyed tonight's event too, folks. Special thanks to our artist in residence, Keith, for the amazing images that have been gracing your screens, and our graphic designer, Nicole, who put together the amazing artwork. You can check it all out on the Pint AU website, pintofscience.com.au. You can also buy fantastic merchandise like pint glasses, there's a cheeky plug, and t-shirts from previous years as well. So pintofscience.com.au for all your pint information. Thanks once again to all our speakers for tonight. Bella, Maddie and Luke, you've been amazing. Thank you to the audience for asking great questions, because in reality, pint events in any format are all about having a conversation. Hopefully we answered your questions tonight, but if we didn't, keep an eye out on Twitter. I'm sure Maddie, Bella and Luke are going to be keeping an eye out there to answer any more questions that they can on their work. But all it remains for me to say thanks very much for tuning in, folks, for Pint AU Online. My name is Broderick Matthews. It's been an absolute pleasure. Hope you have a good night and keep your eye on the science. Good night, all.
It wasn't that long ago, before the first mobile, that plans were set in stone without relying on a telephone. You had to commit. When you made a plan, that was it. 'Cause there was no way for your friends to reach you if you didn't show. So give me your word, give me a guarantee that what you say will be an eventuality. Yeah, give me your word. Won't you give me a guarantee that what you say will be an eventuality? The times have changed. We don't put a pencil to the page, and words upon a screen are easier to erase. We didn't think about this. We didn't think about what we missed. Now it's easier to excuse yourself than to commit. So give me a word, you give me a guarantee that what you say will be an eventuality. Oh, yeah, give me a word, won't you give me a guarantee that what you say will be an eventuality? Well, if you make plans with that one friend, and when it comes the weekend, you decide to put something else first. Well, you might not think you're you give me your word? Won't you give me your word? You give me your word. You give me your word. You give me your word. Won't you give me your word? Won't you give me your guarantee that what you say will be an eventuality? Oh no, won't you give me your word? Won't you give me your guarantee that what you say will be an eventuality? You're gonna see it'll set you free when you...
Yeah, I don't know what happened to me. Like literally, I saw the countdown